and I am delighted to be here. I am really touched by the help that my lab has gotten directly by perfusionists who have suggested things that we could be doing, suggested measurements that we could make. I have worked now for 40 years with a group of organisms that are waterborne pathogens. So you face them, and humans are surrounded by them all the time. And our patients tell us their own experience, and they guide our research. So our research is not separated from patients. It's not separated from perfusionists now, a word that I simply learned earlier in April that I'd never heard before. So I am new to this, so that please ask any questions. I'm going to be around here for the rest of the day. I have a 7 o'clock flight back to Blacksburg because tomorrow morning I teach. This doesn't look any different from my class. I think if we offered food at 8 o'clock classes, I'd get much better attendance. Nobody sits in the front several rows, and then everybody is hiding out in back. So I feel very much at home, and you've made me feel very much at home here. Well, I want to talk about non-tuberculous mycobacteria and heater coolers. And so let's just move on to first give you a little background about these organisms. They are environmental opportunists. Environmental means they come from the environment. They are not transmitted from person to person. They're opportunistic pathogens, meaning that most of us get exposed, in fact, all the time, but we don't get infected. There are just certain groups of people that have risk factors for this disease. We now know that there are over 200 distinct species of these organisms that are being discovered, and on the way down I read 3 or 4 papers describing new species. So we still don't have an idea of the full scope of this group of organisms. There are 2 groups based on their growth rate. Now, for a microbiologist, many work with Escherichia coli, a human inhabitant that we use for water monitoring, and is a standard, unfortunately, for many things that will impact on heater coolers and disinfection. E. coli has a generation time of 20 minutes. So when I was a graduate student, I could do experiments. I could plan an experiment in the morning, do the experiment in the afternoon, and look at the results the next day. That's linear science. With mycobacteria, their generation time is 24 hours, or a little bit less. That means that my students have to learn to plan experiments, do an experiment knowing that they won't see the results for 2 weeks, so they have to do the second experiment even though they don't know the results of the first. So there's always a long lag time. This has been a difficult problem for many of the clinicians who are dealing with mycobacterial infections, because they're used to sending a sample to the laboratory, they get a result back the next day or the following day, and what we do is we simply say, well, we'll be glad to tell you the results in 3 weeks. Now that bothers a lot of people, so our progress sometimes is held up until we get on this bandwagon of being able to plan and do experiments quickly. We have 2 groups, Mycobacterium abscessus, which is one of the organisms that's been found in heater coolers and linked to aerosol infections. Then we have the slowly growing group, Mycobacterium avium. Don't be confused, even though it's called avium, we're talking about an organism that infects humans. Mycobacterium intracellulare, because these are intracellular parasites. They grow in white blood cells. That's where they are in infected humans. And a newly described relative, Mycobacterium chimera. It's called chimera because it has characteristics of intracellular and avium, and is in fact almost identical to Mycobacterium intracellular. The one key character that drives a lot of the behavior of the mycobacteria is that they have an outer membrane. So they have the cytoplasmic membrane that all cells, plants, animals, humans, insects, all organisms have. Then there's a cell wall. Bacteria do have a cell wall that gives them a rigid structure. Some bacteria have another membrane layer on the outside. The mycobacteria have an outer layer that's very thick and made of very long chain lipids. They can be upwards of 60 to 80 units. That makes them little wax beads that are impermeable to everything, including disinfectants, including antibiotics. It makes them hydrophobic. So rather than floating around in the water, they attach to surfaces. So if we were a water reservoir, I would not be here, you would not be here, we'd all be stuck to the wall. There would be very few floating in the water. If I had Velcro, I could run and stick myself onto the wall, but not today. Here we go. Infection. There's no transmission between individuals. There's been we were worried early on in the AIDS, HIV epidemic where mycobacteria infect late-stage AIDS patients, that by putting AIDS patients together, they would infect one another with Mycobacterium avium. That did not happen. More recently, there was a furor of information that Mycobacterium abscessus was being transmitted between cystic fibrosis patients. Cystic fibrosis patients are exquisitely sensitive to mycobacterial infection, but that transmission has not been really well documented. So we don't think there's any transmission there, even though those patients are extremely susceptible. One of the difficulties that FDA is having now with heater coolers is the question of how many, Joe, if they ask me, are needed for infection in normal people or in immunocompromised people. We simply do not know. There is no established dose response for mycobacterial infection. That means that if you tell me there are 1,000 in a milliliter of water, I'll say that's relatively high for water. Normally they're about 100. But I don't know if that's beyond the threshold for infection. Risk factors: physical lung damage, smoking, working in a dusty occupation. In our part of the country, black lung disease is coming back for some yet unknown reason, and black lung patients are very susceptible to mycobacterial disease. We also have on the North Carolina-Virginia border a large number of textile plants, and people who work in there end up with something called brown or gray lung, which is all of the fibers. Those predispose to mycobacterial disease. Dental surgery. There's a big kind of box next to the chair where you're sitting, and that's where water comes in and the water is sprayed in your mouth. The water stays in those things, and when they spray water into your mouth and you've had dental surgery, you have a very short passage between where the tooth was and where the lymph nodes are, and there are infections there. And there are a number of reports in the literature documenting mycobacterial infections in the cervical lymph nodes. Normally, those infections are found in children who, like my children, I encourage them to go outside and play in the dirt and do things like that. They play in the dirt, they drink things, they put things in their mouth, and that's where we find it in children with erupting teeth. But now we're also finding it increasingly in dental clinics associated with surgery. I said cystic fibrosis is a risk factor. That's another lung predisposing condition. The immunosuppressed, and we have surprisingly a large number of immunosuppressed people because of infection, because of cancer, and because of chemotherapy. We also have a group of older, slender, taller women. The body mass index is about 20. The average height is 5'8". The average age is 55 and above. They make up the majority of mycobacterial infected patients. Mycobacterium avium, Mycobacterium chimera, Mycobacterium intracellularia, a variety. Why those women are more susceptible has not yet been elucidated. A portion of those people, there are a few of the predisposing conditions. They may carry just one mutant gene for cystic fibrosis, not two. They've lived 60 years with no problems, and then they hit that 60 years and they now are susceptible. So these are the risk factors. There are about 80,000 of those slender, older, taller women in the United States. Okay. Point it this way. I'm not going anywhere. Okay, whoops, we missed. Maybe I missed one. Let me go back and see. Okay. All right. That's fine. I'm using different notes here. Okay. The consequences of the hydrophobic outer membrane. They grow slowly, these mycobacteria. But that's because they take all of their energy and they channel it into making these long-chain lipids. Each addition of 2 takes 1 ATP, and you may have remembered that ATP is the energy molecule in cells, so they are diverting all of their energy into that outer membrane, not more cells. But that makes impermeable cells. They grow slowly as well because charged agents such as phosphate, metals, don't get into cells because the cells, they're relatively impermeable. Their transport is about 100 times slower than for normal organisms. But even though that might seem to be a bad characteristic to have, in terms of our engineered environments, it is a great thing to have because the mycobacteria are resistant to disinfectants, chlorine, ozone, chloramine, chlorine dioxide. So they survive in drinking water systems. They don't survive in water that has other organisms. They don't survive in environments where there's a lot of organic matter, but they do love to be in drinking water, and they're happy campers in drinking water. They prefer adherence. If you're a slow-growing organism, if there was a water flow through this reservoir that I proposed that we had here, we would all be washed out through the door. But we wouldn't be washed out if we were attached to the wall. Mycobacteria are normal inhabitants of rivers and streams, which are flowing systems. They stick to things. If you want to find mycobacteria, you can find them really readily down here. Don't walk towards the ocean that way, but walk in the inland waters, in the estuarine inland waters, take a little Q-tip and swab the roots of one of the mangroves. You will find a population of about 1 million mycobacteria per square centimeter, which is a pretty small area. They love estuaries, but they also love drinking water systems. They grow on low organic carbon levels. Now that too is a characteristic of drinking water. As a matter of fact, EPA about 10 years ago said you need to reduce the organic matter content in water because chlorine plus organic matter makes carcinogens in water, and that's one of the big EPA pushes right now. Well, again, that then selected for the mycobacteria and gets rid of their competitors. So they're in drinking water and they're perfectly happy because they no longer have any competitors for the limited amount of organic matter that's there. They grow under reduced oxygen conditions. So when you left your homes perhaps yesterday or the day before, you, like my wife and I, we get up and we shower, we fill the house system with warm water throughout. Oh, that's good because warmth means higher growth rates. But the organisms consume the oxygen, so the water becomes stagnant. The oxygen level falls. Mycobacteria can grow on about half at normal rates with half the oxygen that you have in air. So they're happy campers when the water gets stagnant. Last, and I'll talk about this some more because that's a key with regards to the heater coolers, they are concentrated in aerosols. There we go. Here's a figure that's been drawn, taken from work that my colleague Bruce Parker and I did some years ago by a colleague and an excellent clinician at the National Jewish Hospital in Denver, Colorado, which is one of the centers in the world for mycobacterial research. Mycobacteria in water, and here they are right down here at the bottom of the figure in red, little red mycobacterial cells adhere to bubbles that are in water. And these bubbles are formed naturally all the time. When the mycobacteria don't like to be in the water, but this air-water interface reduces and allows them to escape dealing with highly charged water because they're so hydrophobic. When this bubble gets to the surface, it bursts, makes a little crater. That crater results in the ejection of small droplets. The mycobacteria are concentrated into those droplets. If, for example, you have air flowing across this, those droplets will be entrained and transmitted in the air. If you have a surface above it, the mycobacteria will end up now being ejected up to that surface where they'll stick and grow and form a biofilm. Can we push it forward? Sorry. Now, what do we have as sources for mycobacteria? And I'll get back to the aerosolization story as we go on. We found them in natural soils and water. If you go to, and here, sadly, and I apologize, I don't ever expect to be invited back to another meeting, very simply because I'll tell you some awful things that surround you. Just be aware of the fact that if you don't have those risk factors, you're okay. So we found them, mycobacteria, in women out in National Jewish group of patients, and they were all gardeners, and we got the idea that maybe the mycobacteria were coming from their garden soil. Because when you work with garden soil, you make aerosols and you can inhale that dust. Well, that was the case. What we discovered further was that potting soil. So the next time you go to one of the local do-it-yourself stores and you get a bag of potting soil, and right now in my home we have 3 bales, literally bales, my wife is a serious gardener, filled with peat moss. Mycobacteria, one of their natural habitats is peat, sphagnum vegetation. That's the stuff that's harvested, dug, put in boxes, added to poor quality soil to enrich the organic matter. And when you get even the smallest of bags of potting soil, per every gram of potting soil, there are about 1 million mycobacteria. They're in natural waters, commercial potting soil. They're in drinking water distribution systems. In fact, the mycobacteria grow in the drinking water system from the treatment plant to your home. Again, they have no competitors. There's organic matter. The chlorine concentration doesn't bother them. There are no competitors, so they grow. They're in premise plumbing. Premise meaning a hospital, a home, a condominium, an apartment, and other places. They love it. It gets warmed regularly. It accumulates organic matter. One of the big places where mycobacteria grow are in water heaters, anything that heats water. But primarily our glass-lined big thermoses that we think of as water heaters harbor very high numbers of mycobacteria. You can drain the hot water heater. That gets rid of the sediment, which has got a lot of mycobacteria in it, but you're not going to be cleaning the biofilm on the glass, and mycobacteria do adhere to glass, and so you can refill it, and then the water will get colonized from the cells in the biofilm. I got a call one day from an engineer in Clearwater, Florida. Very nice guy, very bright guy. He said, "My mother and father have mycobacterial disease. Could you sample our house?" Their house. And that was when we had grant funds to do that, and we were interested, so this was an opportunity. And one of the few cases where we had both a husband and wife that had NTM disease, both taller, slender, older Americans. Well, one of the samples yielded a petri dish with about 10,000 mycobacterial colonies, and my technician, Myra Williams, said, "Joe, look at this." I said, "That looks like a pure colony of Mycobacterium abscessus." And she said, "I think it is." And I said, "Where did this come from?" And she said, "It came from their refrigerator water tap." Now, in the United States here, we have nice big refrigerators, and you can get ice and chilled water from your refrigerator. Well, quite frankly, folks, rip it out. It has a tank, the water gets warmed, the mycobacteria love those systems. In this woman's case, who was the first one to come down with disease, her physician had told her to drink 6 8-ounce glasses of water a day. She did. She wanted it chilled. She got it from the refrigerator and was getting probably 10 to 100 million mycobacteria in every glass. So if you drink 100,000 mycobacteria every day and you have gastric reflux, you're going to come down with pulmonary disease. I can tell you at that end, that's a reasonable dose response. Hot tubs, another thing to get rid of. Spas, therapy pools, they all generate aerosols, and remember, the mycobacteria are concentrated in those aerosols. Humidifiers. We have a new group of humidifiers out there called ultrasonic. They generate the aerosol by the vibration of a ceramic disc. The one that I remember exposing my children to had a little paddle that splashed. Okay? But that aerosol from those kinds of humidifiers has exceedingly high numbers of mycobacteria. Even if we only put in, and we did, 100 mycobacteria per milliliter, which is a kind of a low normal standard, we got 20,000 per cubic foot of air, putting it into a room that's about the size of a bathroom where we do some aerosol measurements. Shower heads. I got a call from a fellow at National Jewish Hospital, Mike Eisman, who is the senior person in the world in terms of mycobacterial disease, terrific clinician. And Mike said, I got a call from a patient. The woman has mycobacterial disease, and she thinks she got it because she had bronchitis, and she thinks she got it from her shower. So I learned two things. One is that many of the treatments for bronchitis and other pulmonary diseases is to give some of the antibiotics that literally get rid of all organisms in the lung. You essentially sterilize the lung transiently. As you almost do when you take penicillin, you transiently sterilize the GI tract. Now, that can result in problems because as we now understand and are growing to understand, the normal flora organisms that you have in the lung, the stomach, the GI tract, those protect you from disease. Well, she had 2 bouts of bronchitis. The drug sterilized her lungs. Because there was nothing protecting her lungs, no organisms, she came down with Mycobacterium avium pulmonary disease. And the fingerprint, the DNA fingerprint, and we can do DNA fingerprinting with Mycobacteria as we can with other organisms, the DNA fingerprinting of her isolate from her lungs was the same as from her shower and hot and cold water. Norm Pace, a member of the National Academy of Sciences, heard me talk about this shower case when I gave a lecture at his institution at the University of Colorado in Boulder. He sent his students out with pipe wrenches all over the United States. They unscrewed shower heads. They took a single swab and swabbed the inside of the shower head. They then sent all those back to their lab, and Norm, who's a DNA-based identification guy, isolated the DNA, and 70% of the shower heads in the United States have mycobacteria. So when I write a paper and the title is Surrounded by Mycobacteria, I'm fairly serious that that's the case. So we have them everywhere because we all come in contact with these. Oh, I need to go ahead. Whoops, I think we did 2. There we go. Premise plumbing. Now, first, before I talk about premise plumbing, I want you to understand that I think that a heater cooler is just like premise plumbing. It has pipes, it's enclosed. So what are the characteristics? It may have a disinfectant. The disinfectant kills off everything but mycobacteria and a number of other microorganisms. The NTM are relatively heat resistant. I'll talk more about that later. They grow on the available organic matter. There's not much in premise plumbing or a heater cooler, but it can accumulate. There's a large surface area for biofilms. Small pipes have a huge surface to volume ratio compared with the pipes that our water is delivered, which could be an 8-inch main going down the middle of your street. Regular warming of the water occurs, both in a heater cooler and in premise plumbing. Mycobacteria and several others of the waterborne pathogen group grow just fine in stagnant water. They also grow in amoeba. And we don't like to think about this, but there are amoeba that populate our drinking water system. Normally they go around and eat other organisms and kill them, and that's how they grow. The mycobacteria get swallowed by the amoeba, and the mycobacteria grow in the amoeba. Legionella gets swallowed by amoeba, and they grow in the amoeba. Pseudomonas aeruginosa gets swallowed by the amoeba, and they grow in it. These bacteria have learned to live with these organisms that eat them, and they've spun it around so that they use them as hosts. Next slide. Now, there are many more mycobacterial cells in the biofilms on the surfaces of pipes in the home, pipes in a heater cooler, than are in suspension. When people tell me they want to look for mycobacteria, I tell them don't collect large volumes of water because the numbers are fairly low. Go around with swabs, swab the end of a tap, swab the inside of a shower head, aerators on faucets on taps. Unscrew the aerator and swab it before you throw it out, because they harbor large numbers. So we're looking at maybe 100 to 1,000 in normal drinking water numbers of mycobacteria in water, but on pipes about 10,000 per centimeter squared. I once calculated the number of centimeters squared for the Los Angeles Municipal Water District, which has 5,000 miles of pipe. There were more zeros than I was used to handling, but we were getting hundreds of trillions of mycobacteria represented by the biofilm just in the pipes, not the water. Cells in a biofilm are embedded in a matrix. First, the cell binds, but then it accumulates extracellular material. It can be polysaccharide, sticky long-chain sugars, lipid in the case of mycobacteria because they have a lipid membrane, but that lipid gets literally exported and is there independently of the cells. There is DNA. DNA is another polymeric material. It's very long and it has high viscosity. And there's protein there. That makes a layer that makes the cells in the biofilm impervious to disinfectants. The adherence is good for the mycobacteria because they don't get washed out, but as I said, they're impermeable to disinfectant. So you can disinfect, you can kill all the water in suspension, and you won't find any, as we have done. You wait a week, you sample again, there's still none. Ooh, we're doing well. Sample again, there are none after 2 weeks. After 3 weeks, there are none. After 4 weeks, they reappear. We haven't inoculated it at all. It's that it takes that length of time for the biofilm to essentially recolonize the cells in the biofilm. Next slide. Now, I want to tell you a little bit and go into some technical EPA jargon here in talking about disinfection. There's a number called CT99%. It's the product of the disinfectant concentration in parts per million multiplied by the duration of exposure in minutes to kill 99.9% of the cells. That's a standard that's used in the drinking water industry. NTM, as I said before, are disinfectant resistant. Here I want to compare, down here in this little table, Escherichia coli, which is the standard for performance of disinfection. Normally, the concentration of disinfectant used in U.S. water plants is about 1 part per million. And the CT value for E. coli is 0.05, which means that in about 3 seconds, at 1 part per million, 0.05 of a minute is 3 seconds, you can kill 99.9% of the E. coli cells, which is our standard for clean water, in 3 seconds. That's why you can have a treatment plant that manages millions of gallons a day and runs water through at a very high rate because you only need 3 seconds to kill E. coli. Well, what happens to mycobacteria? Mycobacteria, frankly, are sitting there, and I really do characterize these things as being living little critters, and they're aware of what's going on around them. They're sitting there waving goodbye to everybody else. Sorry, you're going to die now. Everybody's gone. Yes, we're the only ones left because their CT value is, depending upon the strain we've looked at, between 100 and 200. So they are so much, 1,000-fold more resistant to chlorine. So chlorine is just kind of, okay, that's something there, but it doesn't bother them. Then, on top of that, you have the mycobacterial cells in the biofilm. They're 5 to 10-fold more resistant than the cells in suspension. Next slide. All right. Here is my analogy and comparison between premise plumbing and heater coolers. Heater coolers are like premise plumbing. Heater coolers have a large surface area for biofilm formation. They heat. They have low oxygen at times. There's organic matter and water and metals that they get from the water. Those things create growth conditions. The source of water in the NTM, in the heater coolers, is the hospital water. What's the quality of the hospital water? Now, I mention that because you can control that. You yourselves can control that. One of the common misconceptions, and I learned this after I got my degree at Berkeley. I was an ROTC cadet. It was 1969, actually. That's when I was commissioned. In 1969, I was in the United States Medical Corps running hospital labs. I did not know what was going on in there, and they gave me an internship in clinical pathology, which I treasure to this day. Taught me some really good stuff that I continue to use. Well, one day there was a problem of some solutions getting contaminated, and we traced it back, and we talked to the people, and they were using distilled water to make those solutions that they were using directly on patients, and they said, well, we use distilled water, and distilled water is sterile. It isn't. That's why I put it in red. Distilled water, even though the water is boiled and ought to kill things, the vessels that we put distilled water in are not sterile. They accumulate microorganisms. Deionized water, that too is not sterile. So, if you really want to make sure, you want to use autoclaved water, 15 pounds pressure for 15 minutes in an autoclave. Hospitals have autoclaves. Or you can filter it. The filter pore size you need is 0.2 micron or smaller because bacteria can't get through that. Bacteria about 1 micrometer in size. Activated charcoal filters actually are good sources of mycobacteria. Again, I'm going to ruin your day. If you use these activated charcoal filters, they get rid of chlorine, they get rid of metals, the water tastes better, that's fine, but the mycobacteria grow, and eventually the activated charcoal filter serves as a source of mycobacteria and other organisms. So that's a key characteristic of pore size. Disinfection may not kill all biofilm cells, so where we have mycobacterial infections in hospitals or in hotels, as a matter of fact, the organisms always come back because they're in biofilms. This is also characteristic of Legionella if you look at the Legionella literature. Biofilm cells are not accessible to cleaning. And here's a word that troubles me. Sometimes I hear people talking about cleaning, and I don't know whether they're talking about scrubbing a surface or actually disinfecting. So I'm always careful about words like cleaning because I'm not sure what you're doing there, and also about filters because if somebody says, oh, I have a filter in the line, you would think something is not going through. But as we've learned and been taught by the granular activated charcoal filters, it's pore size that's important. Next one. I've talked about aerosolization, but I want to talk about it in terms of heater coolers. That the water reservoirs, air bubbles are continually rising. Even though the machine is off, you can take a Petri dish and hold it 10 centimeters above the surface of a water sample, and the little ejected drops will end up on that. You can actually then incubate that and see the mycobacterial cells. You do it with Pseudomonas aeruginosa. You can do it with Legionella. Okay. The mycobacteria are concentrated into the droplets. The droplets are small enough to be entrained and move away in the air. Next slide. Here is a picture taken by Duncan Blanchard, who was an atmospheric physicist at the State University of New York in Albany, who taught us this. The picture goes in the following fashion. Here's an air bubble reaching the surface of water, the air-water interface. Here's the bubble is now bursting. Here's the little crater formed. Now, if you remember physics, and I learn everything through microbiology. I've learned chemistry. I've learned everything through microbiology. Here's physics. There's a force vector here that wants to close. There's one from the other side, and there's one from here. These two force vectors are balanced. This one is not. So there's a droplet. There's a droplet. That's the little droplet that gets entrained and can be as high as 10 centimeters above. Next one, please. In terms of heater-cooler disinfection, I am slowly learning something from the industrial scientists and the hospital engineers. Hazard analysis critical control points. Can't even say it very well. What are the critical components, critical control points? Now, control means where you can exert your activities. First of all, the addition of sterile water to a heater cooler. You put in drinking water, you're inoculating the heater cooler with microorganisms. You can follow the cleaning and disinfection manufacturer's instructions. Those have been worked out, measured, vetted, but you have to follow those. How frequently? In the absence of any guidance, you need to clean and disinfect monthly. Remember, I said when we disinfect, the reappearance of mycobacteria takes about a month. So that way you keep the numbers at a very low rate. You can monitor the non-tuberculous mycobacteria in the reservoir water. You can collect a sample. The lab can take a volume of that, spread it on medium, and tell you what's in there and what's growing. They don't normally, a clinical laboratory doesn't normally handle water samples. But once it's on a petri dish, it's no different than a throat culture, a sputum culture, a blood culture. You want to get low numbers. Now, FDA is really worrying about what kind of numbers, because there is no guidance. It would be crazy to think that a heater cooler could be sterile. As you all know, it's just going to be impossible. Can you completely disinfect a heater cooler? No, you cannot. If I could disinfect a heater cooler, I would love to do that, because then I could do experiments like as we do in the lab. I start with sterile tubes, I start with sterile stuff, and I don't have other organisms, and I could look at just mycobacteria or just Legionella or just Pseudomonads. But I can't do that. I have to deal with a population when we look at a sample from a heater cooler. So we have to kind of work around some difficulties and barriers. So we're always going to have some level of contamination. What we need to do is to provide you with guidance on keeping those numbers as low as possible. And that's where, in a way, I'm starting out here. Let's say the next slide, which may not be another slide. No. Well, it's 5 minutes to 9. Normally I run over, but I have 5 minutes now. I'll be happy to answer any questions now in the next couple of minutes, and I will also be around here for the rest of the day. My plane is 7 o'clock. I think I'm being picked up at 5 o'clock to go to the airport, and if anybody wants to talk to me, I will wander outside here. I don't want to take away from your attendance at the rest of the meeting, but I'll be more than happy to answer any questions anytime during the day. So, thank you very much.